Hey class, so this is my sixth lesson, and this is from lecture 4 PowerPoints, which is on translation and proteins. So hopefully a lot of this is review from your intro bio class. If it isn't, then I see some extra studying in your future. So remember that the RNA has snaked out of the nucleus and eukaryotes, and prokaryotes is still in the cytoplasm. And that is going to be read by ribosomes, and ribosomes are going to take the order of letters in the RNA and convert that into amino acids that form proteins. So just to remember, there's 4 letters in RNA, and there's 20 different amino acids. And also, I want you to remember that amino acids have different chemical properties. Some can be positively charged, some are negatively charged, some are not charged. Some are acidic, some are basic, some are hydrophobic, which means water-fearing, and some are hydrophilic, which means water-loving. So it's those chemical properties that dictate the shape of the protein, and the shape of the protein dictates its function. Like most mutations and the mutations that I study, like sickle cell anemia or cardiovascular disease or cancers or whatever, it's these misshapen proteins that either lose their function or gain a function that isn't the proper function. Most of the time they lose their function because they don't have the right shape. Kind of like trying to put a square peg into a round hole. So just remember, proteins are called polypeptides. It means the same thing. It's just a different word, just like cash and money or moolah or dinero or whatever, bones, whatever you want to call it. So when we say polypeptide, we're talking about a protein chain. That protein chain is made up of monomers. Those monomers are amino acids. There are 20 amino acids, and they have different chemical properties. The bonds that join these amino acids together, these monomers, to make the polymer that we call a peptide or polypeptide or a protein, is called a peptide bond. And proteins have lots of different functions, right? They can be structural support, like keratin that you find in your nails, or keratin is also what is in the nucleus that maintains its structure. You can use it to store things like albumin in your blood. So that's a source of amino acid storage. Same thing, chicken egg whites. To transport, so hemoglobin is a protein that transports oxygen. It can be signaling. So insulin is a hormone, but it's a protein hormone. And so insulin is a protein. The receptors in your eyes that respond to stimuli, those are made out of proteins. You could have a mutation in there where it doesn't function properly, and then you could get color blindness or other things. It's responsible for movement. So myosin is the motor protein that moves your muscles. Your immune system is made out of protein. So the antibodies that you generate from your B cells are made out of protein. And then, of course, we know that enzymes are proteins that are catalysts that speed up reactions. Okay, so like we said, the monomers for proteins are amino acids. And again, hopefully this is a review, but there is a central carbon, which is shown here, and there are always 3 groups on this central carbon. So we have the amino group, and then we have the carboxyl group. So if we are, okay, so I apologize. I'm using my office computer. Usually I use my laptop. And the audio may be different too, because I'm not sure about this microphone. But anyway, as I was saying, here's the draw tool. We have a central carbon. And we have an amino group on one side. So amino, in case you don't remember your functional groups, is NH2. So this is an amino group, which is shown here. And then we have carboxyl, so C double bond O to O to H. And this is also known as carboxylic acid. So that's where the word came from, amino acid. It's the functional groups on either side of the central carbon. And then there's always a hydrogen on one end, and then we have the R or variable groups here. And like I said, there's 20 different ones. So I don't expect you to memorize those. You can look them up on a table. But what you should know is what properties they have and how that's going to function. So let's say that we have a protein chain and we have two positive R groups next to each other. So this is the backbone. Let's just say that that's an amino acid. So what do you think is going to happen to the shape of this protein if you have 2+ charges next to one another? It's going to make it fold away, right? The R groups are going to want to repel from one another. So they end up far away. So that you might have a protein that bends this direction. Now, let's say you have a mutation which causes this positive R group to become negative here. Now they're going to attract. So it might bend this way where the R groups are attracted to one another as opposed to repelling one another. And so that would cause it to have a different shape, right? You might want to have a localized reaction, like the reaction to link amino acids requires, and again, hopefully this is a review. So to link amino acids to form long chains of amino acids requires a reaction called dehydration. And so you want to remove water. So you would want these R groups to be hydrophobic, right? Because you don't want to pull in water, you want to release water. So that would help water to be removed. If you wanted to split them, so breaking apart of macromolecules is hydrolysis. And in that case, you would want these to be water loving, right? Hydrophilic. And that would bring water in to this bond. And remember, what's this bond called? Peptide bond. Water comes in and breaks that bond because the R groups are attracted to water, right? So you're making a little microchemical environment. Anyway, so that's the shape of amino acids and the function of amino acids and how they're built and broken apart. It's kind of interesting because my wife was asking me about collagen protein versus whey protein versus other protein, for increasing your macros for protein. And, there are commercials on TV talking about how collagen's important for you and you should buy collagen amino acids and blah, blah, blah. And I was like, that's really dumb because what happens as soon as you swallow proteins, the acids in your stomach break all of these peptide bonds. So collagen is in a specific order. If it's chopped up into individual amino acids, your body has to reassemble it after you digest it. So it doesn't, it because it came from collagen doesn't mean anything. It just means that the ratios of amino acids are different, but it doesn't mean you're going to magically make collagen. All right, so peptide bonds, and we talked about dehydration reactions, right? So it's a covalent bond here formed by a dehydration reaction, which is shown here. So we have a hydroxyl group, hydrogen on the amino, the hydroxyl on the carboxyl group. Here's the amino group. And then those, we remove water, and that links them together. So we have a new peptide bond. And then we can continue on. This is the reaction that the ribosome does. So remember, we have these are always the same. Hydrogen, amino, carboxyl. Amino, hydrogen, carboxyl. So this just repeats over and over again. So we call that the backbone. It never changes. It's always the same. These side chains are the R groups, and they vary. Those are variables. So this one's aromatic. This one's can form disulfide bonds that are really important. This one is the hydrogen can leave, so could be negative. Same here. This is ring structure is made out of carbons and hydrogen, so this is going to be hydrophobic, right? Because again, this should be review, but carbon's electronegativity is 2.5, and hydrogen's is 2.1. So we take the difference, that's 0.4, and that difference in electronegativity equals a non-polar covalent bond. So if it's non-polar, water can only surround things with charges. So if it's non-polar, then it's not water soluble, and therefore it's what we call hydrophobic, right? It's afraid of water. Okay. Next slide. This is just an animation of synthesis, and I don't think it actually works anymore because Microsoft changed their macros to because everything is infected with the virus now. So some hacker ruined it for everyone. Okay, so protein configuration. The most important thing in biology, you get nothing from this class, the most important is shape. Right? I study all kinds of diseases, and I can tell you that 99.99999% of them are caused by a misshapen protein. So, what determines the shape? Well, it's the three-dimensional structure of the protein is determined by the sequence of amino acids in the protein. So you have your DNA, right? I'm just going to draw it 3 to 5, and you guys did a bunch of problems on this. And so the RNA would be, no polymerase can do anything but 5 to 3, like we said. This is the messenger RNA. We talked about it gets processed or whatever, and then it goes out to the ribosome. And then that reads the mRNA and turns it into a protein. So, and it's the order of these letters, like the order of these letters determine what amino acids the ribosome incorporates and in what order. And like we said, if they're too positive, they might be repelled. Two negatives, they might be folded together. If a hydrophobic is next to a hydrophilic, they're going to push away, and so on. And so that all of these interactions of these amino acids and their chemistry cause them to fold in specific shapes. And so there's two general shapes that we can look at when we're looking at the images. So this is what we call the space filling model. This accounts for the orbitals of the atoms. And so you can kind of see, like, maybe there's a pocket right here. that a molecule might fit in. This specific shape might be a shape that fits into DNA. So looking at these space filling models kind of gives scientists an idea of what its function may be. This is beneficial because we can't see the inside of here, but we can see a little bit of this sulfur here and here because it's yellow, but I can't see any of it on this side. So this ribbon diagram is useful in just showing me where the disulfide bonds are, which are here shown in yellow. Where are the beta sheets, which are shown here because they're flat. Those are interacting with each other. And then this is an alpha helix right here. And so I know that this is an alpha helix. And this is a beta sheet. And these are disulfide bonds that are going to give it rigidity and structure. Since this has so many disulfides in it, it's probably a protein that's not inside the cell. It's probably secreted by the cell. So maybe it could be like amylase, which is secreted in your saliva to break down sugars. It could be like an enzyme that's designed to break down bacteria cell walls in your eye, and so on. So anyway, just remember that this is the ribbon diagram, and this is space filling. All right. So I can't stress this enough. The most important thing in biology is shape. Like I tell my 181 students all the time, I'm like, if you're going to make a cologne that mimics a pheromone, what would be the most important thing you look at? And that's the shape of the pheromone. So, same thing here. This is a DNA polymerase right here. We talked about that, right? It's the thing that copies DNA. This one is polymerase 3. And of course, because it's a 3, it's in a prokaryote. And then this is DNA, right? DNA structure is the same for every organism. This polymerase has a specific shape. It binds on, like you can see how it's hugging this DNA specifically and precisely. And if it didn't have this shape, it wouldn't fit on the DNA strand. So the polymerase shape dictates that it's going to interact with DNA itself. And that could be for anything, right? Binding to something, causing it to change its shape, causing a chemical reaction, causing it to perform a function. All right, so we have 4 different levels of organization for proteins. The primary, secondary, tertiary, and quaternary. You probably learned this in intro biology, but we're going to go over it again because it's super important. So, quaternary structure, not all proteins have a quaternary structure. Some proteins stop at tertiary. Quaternary is only if you have 2 or more proteins necessary to form a final protein product that makes it functional. So I'm going to show you this later, but so collagen, like we talked about, that they're trying to sell you to increase your collagen, that's not going to do anything. That is requires 3 collagens. They're intertwined, kind of like this, and they give it shape. So we have 3 collagens that make it functional. 1 collagen wouldn't be strong enough, so it wouldn't be collagen. Hemoglobin is another example. So hemoglobin is made up of 2, I'm just going to draw circles. So 2 alpha and 2 beta hemoglobins. By themselves, they wouldn't be able to bind or carry oxygen because the heme, the iron, is carried in the middle of this. So hemoglobin has a quaternary structure. Insulin is actually two different proteins linked together, so that has a quaternary structure. Amylase doesn't, so it doesn't have a quaternary structure. All right, let's see. And remember, all this stuff comes from genes. I don't really need to say that. And it's the order of the letters in the gene that orders the letters in the RNA, which orders the letters for how the protein is going to be built. All right. So primary structure is simply just the sequence of amino acids, like letters in a book, right? Rat, cat, fat, whatever. The primary structure is always numbered, and this is so that scientists know what we're talking about to one another, even if we're speaking in a different language. Like if this is a threonine, for example, if there's a mutation here, and let's say threonine 70 became proline 70, I would know that mutation occurs exactly here. And that number is dictated by the numbering system. So we always number from the amino to the carboxyl end, right? Just like, remember we were talking about the DNA is always written as the coding strand. And that's always written in the 3' to, always written in the 5' to 3' direction. Same thing with amino acids. They're always written, they don't have, so remember DNA, I'm just going to recall this. So this is the number 1 carbon, 2, 3, 4, and number 5 carbon here, and that's connected to the phosphate. That's an O, by the way. I ran out of room. There's a phosphate here. This is where the base is. Remember, it's either a purine or pyrimidine. So that it dictates whether it's 2 rings or 1. Because it's DNA, there's a hydrogen here, and there's a hydroxyl in this end. If this was RNA, there would be an OH there. But we're not talking about RNA right now. Okay, so amino acids are always linked in this direction, always from the amino. No, let's see if I can spell today. Amino to carboxyl. Because it always has an amino end and a carboxyl end. I'm just going to go back to this here and just remind you. So it can't add on this end, right? It can't add on the amino end because it has to take out water. It has to take out H2O. So it can only add on this hydroxyl end. It's the same principle as it can only add on the 3' end of the DNA because that's the end that has the hydroxyl group. So it's always built from amino to carboxyl because this has that OH or hydroxyl group. Same reason for this one. DNA polymerase can only go 5 to 3. Okay, so you don't want to number starting here and go 1, 2, 3, 4. That would be completely wrong, and we would end up with this completely separate thing. And so if someone were out telling me this and they numbered it backwards, I'd be like, what are you talking about? So you just got to get used to numbering it in this order, in this direction. Amino to carboxyl, always. Determined by genes, we know that. Can be sequence, so we can sequence proteins just like we can sequence DNA. It's a little bit trickier to sequence proteins, but it can be done. And then small changes in any one of these could have a big effect, right? Because we're talking about something that's going to change its shape or could potentially change its shape. We'll talk about mutation in the next chapter and how some mutations are worse than others. But for right now, let's say we have a really big change. These are two chemically different amino acids, glutamic acid and valine. That's going to change the structure. So this is a hemoglobin, one of the subunits of hemoglobin. And what happens is that because this hemoglobin changes shape, it can't load oxygen properly. And if it can't load oxygen properly, kind of like filling up a balloon, right? If you breathe a bunch of oxygen inside this cell, it would have a nice round shape. And you want that, right? Like a nice round cell is going to roll through your bloodstream. This is my blood vessel drawing. But there's not enough oxygen, so it would make your balloon kind of collapse. And in this case, it collapses into a sickle shape. That didn't work out. Collapses into a sickle shape. And so now you have these sickle cells going through your bloodstream, and they catch in here. And like we talked about this before, like you can have these pleiotropic conditions from sickle cell. And it's all caused by one single mutation in one single amino acid that converts glutamic acid into valine at the number 6. So this, I know, is which end? Amino. And this end is carboxyl from the numbering system. Right. So small changes can have big effects, and we'll talk about what the chemical differences between these two and how mutations arise and how some are worse than others in the next lecture series. All right, so that's primary structure. It's simply just the order of letters of amino acids. The second level of organization we're going to talk about is secondary structure. So if we look at the ribbon diagram that we talked about before, this is the same molecule, by the way, and I drew the beta sheets and the alpha helices here. So beta sheets are flat. Alpha helices are helices, just like the name implies. So we have the amino and carboxyl ends of each of these. So this is amino, carbon, carboxyl, amino, carbon, carboxyl, and so on. And so remember, hopefully this is a review, but when you have nitrogen and hydrogen, the electrons stay around nitrogen a lot more than hydrogen. That gives this a partial positive charge. That gives this a partial negative charge. And that's what that signifies. So if you have something with a negative charge and a positive charge, they're going to be attracted to each other. And same thing occurs with oxygen. So these little dashes right here are hydrogen bonds that occur from these partial charges of each of these molecules. Oxygen's very electronegative, so it's going to keep the electrons around it a lot, so it's going to be partial negative. Hydrogen is very weak electronegativity, so this is going to be partially positive. So these sheets form hydrogen bonds, right? And so I'm going to tell you that these are the exact same number of amino acids and the hydrogen bonds that are formed here. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 H bonds in beta sheets. Again, this is the helix. Here's the same number of amino acids. 1, 2, 3, 4, 5, 6, 7, 8. So 8 H bonds. So this, because it has twice the number of bonds, is about 2 times as strong of a structure as a an alpha helix. So beta sheets are about twice as strong as alpha helix structures. These are more flexible. I'm not saying that they're not important because you can respond to your environment better. These are more rigid, so they're not going to change much. And you remember your body's exposed to all kinds of different things, different temperatures, different pHs. And so you kind of have to decide if you want to allow your protein to flex a little or you want it to be more rigid. And so these disulfide bonds make it more rigid, beta sheets make it more rigid, alpha helices make it a little bit more flimsy. And those are the two structures. So these are formed by interactions in the backbone. Right? This has nothing to do with the R groups. Okay, so let's back up. This right here has to do with secondary structure. And it's formed by hydrogen bond formations in proteins. None of this stuff has to do with the R groups. This is just that repetitive amino, carbon, hydrogen, carboxyl group. And that produces alpha helices and beta sheets. So beta sheets are super strong. It's quite interesting. I'm going to talk a little bit about this and show you like a quick video. But so silk, and we're talking about spider silk, not silk that comes out of silkworms. The structural property of the strength of silk is due to beta sheets. Lots of beta sheets. In fact, spider silk is almost all beta sheets. So because of all those hydrogen bonds and all those beta sheets, silk is actually stronger than steel by weight. So you can use it for a lot of stuff. It's stronger than Kevlar. So you could use it for, military purposes or like bulletproof vest purposes, or even like lining the inside of tanks or Humvees. There's a lot of other crazy stuff you can do with it too. If you think about it, like anything you could use steel for, you could use spider silk for. So you could use this to build buildings. One of the craziest ideas is that you could take the Earth and send up a satellite that's in the geosynchronous orbit. So that means that it rotates the exact same speed as the Earth. And then you get a cable that's made out of spider silk. And then, the space is about 100 miles. So this cable will be about 100 miles. It'd be about the distance from, here to Tucson or something like that. And then you can make an elevator. So that elevator would just travel up and down that silk cable made from spider silk. Okay, so I think, hopefully I've convinced you that spider silk is good and it's important, but spiders are not social creatures. You can't just go, hey, spider, spin me a web. They're not going to do that. And so it's very hard to collect a lot of spider silk. So I'm going to show you how hard this is. So I'm going to Okay, I'm going to move this over here. Hopefully it picks this up. But anyway, so all right, so here's an example. So they did this as an experiment. They took spider silk and they collected it to make this cloth. So 70 people spent 4 years collecting spider silk from golden orb spiders. These are Madagascar spiders. They collected them from telephone poles in Madagascar. Another dozen workers extracted the silk, and so they made this. It's 11 feet by 4 feet. It's the only large piece of cloth ever made from natural spider silk that exists in the world today. Those are the spiders that made it, and so this toxin's incredibly strong compared to steel or Kevlar. So there's a purpose for this, right? But it took all that time and all that effort just to make an 11-foot by 4-foot cloth. So how long would it take to make a 100-mile cable or what have you? And so that would be theoretically impossible. It would take, tens of thousands of years to pull that off. But this is the beauty of genetics, right? So remember, the genetic code is universal, which means that you can take any gene from any organism and put it into any other organism. And so, ta-da, spider goats. So research at the University of Wyoming took the gene for spider silk and put it into goats, and in their milk they secrete this protein, and if you put it in a centrifuge it will self-bind. And so in this instance you could make spider silk from goats, and they're doing that right now. So if you want to go and visit them, you can. If you look on YouTube, and the first spider goats were named Pudding and Sweetie. If you want to go meet them, you can just travel up north a little bit, and they're there producing spider silk in their milk. So that's kind of cool. And the reason that we're so interested in this is because of the hydrogen bonds due to the beta sheets. All right. Hopefully you learned something new. All right. So the next level is the tertiary level. This is the three-dimensional final shape of the protein, and this results from the interaction of the R groups. So remember, the primary structure is just the order of amino acids. Remember, it's the amino to carboxyl. It's read in the RNA from the 5' to 3' end. So if we're going to go through this whole thing, we have our DNA, 3' to 5'. This is the template strand, which is also the non-coding strand. And the RNA, the mRNA, is going to be read 5 to 3, right? It can only do it that direction. And then the ribosome starts reading it on the 5' end and goes that way. So we have an amino, and then let's just say it's lysine and arginine and threonine and I don't know, tryptophan and blah, blah. And then we have the carboxyl. And so we have the amino and the carboxyl and then the order of the amino acids. So that's the primary. And then secondary is the interaction, hydrogen bonds in the backbone only. And then we have the tertiary, which we're talking about now, and that is the interaction. There's lots of them, interactions of the R groups with one another. So it could be a hydrogen bond, which is shown here. So two R groups interacting. We could have ionic bonds, which is shown here. Hydrophobic, which is here, right? Carbons and hydrogens, they don't share equally. So these are non-polar bonds. Carbohydrates are hydrocarbons, both of those same bonds. And then we have disulfide bonds that we talked about, right? That helps give protein its rigidity. So all these things can occur between the R groups, and that helps it fold into its final shape. And then we talked about quaternary structure. So some things have quaternary structure and some don't. Like I was showing you before, collagen is 3 strands. It's not functional without those. So because it has more than one peptide, right, protein, that's required to make it functional, then it has a quaternary structure. We talked about hemoglobin. We have alpha and beta chains of hemoglobin. We need all 4 of these to make it functional. Any of these are missing, it's not going to work. It won't have the right shape. So because there's 4 subunits to hemoglobin, that is also considered to have a quaternary structure. All right, so again, here's the primary structure. It's just the order of the amino acids, whatever that is. Remember, it's carboxyl or amino to carboxyl. The beta sheets and the alpha helices, those are interactions of the backbone, and specifically in hydrogen bonding. Which is stronger, beta sheets or alpha helix? Beta sheets, right? We talked about spider silk. The 3-dimensional structure, which is also the interaction of the R groups, is in here, and then quaternary structure, if there is one, is more than one linked together to give it its final shape. All right, so hopefully you guys understand how proteins are folded into their proper shape to make them functional. So we talked about this, like most scientists were team protein because there's 20 different amino acids, right? And so they assume that you could make more complex structures from proteins than you could from DNA, right? DNA, they didn't think was the genetic material because there's only 4 nucleotides. Right? So what scientists finally figured out is that how DNA is able to code for more than 4 amino acids is simply statistics. So if there's 4, like let's say there's 4 letters, we'll say this is DNA, A, G, C, and T. And let's say that there's 4 amino acids. So let's just say lysine and tyrosine and tryptophan and isoleucine. We'll just have those 4. But there's others. We have alanine and arginine, which is a pirate's favorite amino acid. Or get it. Anyway, so we can code for these with 4 letters. And so if there's 20 amino acids, we need at least 20 permutations of that. So if we have the probability of 1 in 4, right, or 4 outcomes, just like we did in dice, we need the rule of multiplication, and then let's say we have 4 more, right? Because if we take this particular spot, this single thing, it could be A, G, C, or T, right? So there's 4 outcomes. So this is the first. The second position could also be A, G, C, or T. So it also is 4. So when we multiply that together, we get 16 combinations. Well, that's not 20. It's still not enough. So if we add another one, then we have 4 times 4 times 4, which is 64. And that's more than we need to code for our 20 amino acids. Because this 3-letter code is required to generate at least 20 different outcomes, right? Because we're multiplying for, in fact, there's 64. Then we call this the codon. So the codon is a 3-letter code, right? It's dictated by statistics and probability so that we can cover all 20 possible amino acids. That codon is in the mRNA. Remember, it's 5 to 3, and we said the DNA is 3 to 5. So this is the template strand, right? The non-coding strand. And so A pairs with U, C pairs with G, C pairs with G, and so on down the line. Right? So this is all correct. The ribosome can read, with the help of transfer RNAs, read this codon, and that codon codes for the amino acid tryptophan. Then the ribosome moves over 3 letters, reads the 3-letter code. UUU codes for phenylalanine. The transfer RNA brings that in. The ribosome moves 3 letters. And so the next group is glycine. I don't know how that just happened. Glycine, and then we have serine here is the next amino acid. Remember, this is the amino end, and then this is the carboxyl end. All right, so that's a codon, and then we have an anticodon found in tRNA. All right, so this is the genetic code. I don't expect you to memorize this. And when I give you an exam, I'll give you this genetic code table. You will need to know how to use it, though. So remember, this is codons, not anticodons. So the genetic code is written in codons. Remember, a codon is in RNA, and we're reading it from the 5' to the 3' end. So we can check the artist who drew this in the textbook, not your textbook, but Chemical Biology is where this is from. And so this tryptophan is UGG. So let's check. So we have the first base is U, second base is G, third base is G. So UGG is tryptophan. And so this artist correctly identified tryptophan as that amino acid that would be put into that codon. Next one is UUU. So let's take a look. And we have UUU, which is here. So that codes for phenylalanine. And so we can check, and yes, indeed, that codes for phenylalanine. And then what's the next one is GGC, and that's glycine. So we can decode it. GGC, and that's glycine. So that's correct. So that's how ribosomes convert RNA, which came from DNA, the 4-letter code in RNA, into the 20-letter code in amino acids. And remember, there's 64 outcomes, so that means that more than one codon can code for the same amino acid. In fact, leucine has 6 codes that code for the same thing. So let's just imagine that you got a mutation here, and that became a G. That wouldn't be so bad because it codes for the exact same amino acid. But let's say you had UUC, and this U gets converted to a C. That wouldn't be phenylalanine anymore. That would be CUC. So we can look C, U, and then C. That would be leucine. So it's a completely different amino acid. So mutations in the third position aren't as bad as mutations in the first. And I'll explain to you why that is, because it has to do with something called wobble, where in the third position, the tRNA doesn't exactly have to match the mRNA. All right. So the reading frame is the frame of reference that you would be reading something in. If we took a words like the fat red cat and we read it out, we know that the frame is the fat red cat. So 3 letters. Now, if we erase this letter and we still read it in 3-letter frames, that would not read the same thing at all, right? So mutations that add or remove a letter instead of changing it can change the whole reading frame. If we took out 2 letters, it still wouldn't make any sense. If we took out 3, then we would lose the word the, but we don't really need that to understand the sentence, right? So the reading frame, mutations in the reading frame, if there's a single mutation or 2 mutations, insertions or deletions that change the reading frame, those are far worse than having 3, because that throws the reading frame back in. And if we just do a little example, here's a sequence of DNA. It could be RNA. So anyway, because it has to be DNA because there's T. So anyway, so they're skipping the RNA step here. But this codes for these amino acids. If we move over 1 letter, it changes all of those amino acids. So reading frame mutations are really bad, especially if they're not in groups of 3. Translation. Okay, so the process of translation is taking those codons that we talked about from the mRNA and converting that into amino acids. So here's the mRNA. Like I said, it's read 5-3, right? This is the mRNA. This is the ribosome. This is the small subunit and the large subunit. These are the transfer RNAs that we talked about in the previous chapter. And then these purple things are the actual amino acids that are linked to the RNA, the tRNA. And those are what are used to read the codon. The tRNA has an anticodon, and then that is what we call a charged tRNA because it has an amino acid on it. So this is charged, a charged tRNA, whereas this one doesn't have an amino acid on it, and we would say that this is uncharged. All right. So we have codons, we have anticodons. This is right here we have UUU pairs with AAA. The AAA and the tRNA is the anticodon, and the UUU is the codon. Okay. And the ribosome directs all of this. So every tRNA binds to only one of 20 different amino acids, right? And again, this is the shape. Shape dictates function. So I don't know if you can see this, but this is alanine. This is the amino alanine. This is the carboxylin, right? This is the amino N. This is the R group, the methyl group here, and then this is the hydrogen. So remember, this is RNA, so this is the 5' end. This is the 3' end. If this is 5, right, we follow this down. It's doing the same thing here. So this is 5, then this is 5, this is 3, this is 5, and this is 3. And remember in RNA, we have a hydroxyl group. So we can link these by taking out water, right? Just like we would a normal dehydration reaction. So you're always going to find the amino acid linked on the 3' end of the transfer RNA. Remember we said RNA is single-stranded, so it can fold on itself, and then you can have this base pairing. So it forms this sort of clover leaf structure, which we were talking about previously. So this is the 3D structure of the transfer RNA that's specific to be charged with alanine. And so the reason it is because it has to have a specific shape, because there's an enzyme that actually charges this, that performs the dehydration reaction required to link the amino acid to the tRNA. So there's, where are we at? Okay, so this is slide 19. So here we have serine, again, the link to the 3' end. And this is the cloverleaf structure.
You'll need to unlock your iPhone.
No, not
You'll need to unlock your iPhone first.
Not use serine. Serine. Anyway, so
you'll need to unlock your iPhone first.
I guess I can't say that, amino acid. So anyway, there is a little bit of wobble room here on this third position. So it really doesn't quite line up. It's more of a lean here. And so these don't pair as strong in the third position as they do in the first and second. So there's a little leeway here where it'll allow a mispairing. And so this would be an exact pairing, right? 3 hydrogen bonds, 2 hydrogen bonds. Same here. These would normally pair, but this is kind of lifted up a little bit. So it allows it to pair anyway, since it's non-complementary. And because of the shape that it is, we call it the wobble. Okay, so here's the rules of wobble. In this drawing, they're kind of showing you that this end is bent up a little bit, and so it's not making perfect complementary base pairs. In this instance, G can pair with U, and it also can pair with C. Sorry, only 3 hydrogen bonds. 1, 2, 3. Thank you. All right, so this is just a table. C can pair with only G, right? C doesn't do this. It doesn't have a wobble. G does have a wobble. Again, this is in the anticodon, right? And then here's the codon. So in the anticodon, if there's a G like shown here, it can pair with C, which is normal, but it can also compare with U, which is not normal. If we have a U, it can pair with an A or a G. Normally it would pair with an A, but it can wobble out and pair with the G, right? So if you get the trend, G can pair with U in the third position, and U also can pair with G in the third position. A can pair with only U's, right? So it's very specific, and we're not going to worry about this. This is a special nucleotide called andosine, and it's super rare. So I'm not going to make you know that. But you should know the wobble rules. So make sure that you guys know those for the test. Okay, so we have the tRNAs. This is the cloverleaf structure that we talked about. Anticodon here. Here's the actual shape. So you can see it's kind of bent right there. And that's showing you the anticodon. This is the ribbon diagram of it. And then this is the artist rendition, which is just for simplicity, which is like a stick of dynamite. So anyway, remember the attachment is on the 3' end because that's a hydroxyl and we had to do that. So there's enzymes called aminoacyl tRNA synthase. The shape of this fits in here, right? So we have to have 20 different ones to fit each of the tRNAs. And remember, the enzymes are specific for their shape. So if we had a different shaped tRNA, or we had the same shaped tRNA, it would get charged with the same amino acid, even though it had a different anticodon, and that wouldn't fly. So you have to have different aminoacyl tRNA synthases to charge these tRNAs. So the tRNA comes in, the amino acid binds. These are linked together. How do we link them together? It's always the same. It's a dehydration reaction, which hopefully this is a refresher. Once it becomes charged, it changes its shape and releases the charged tRNA. And that can go and be incorporated into the growing polypeptide chain in the ribosome. So this is the reaction. I'm not gonna get real crazy specific on this, but this is we need energy to do reactions generally. So the energy comes from ATP. That's turned into a monophosphate. This is a triphosphate. This is an RNA, right? So because it doesn't have a D in front of it, so this is the A letter in RNA. We have A, G, C, and U. So this is it, adenosine triphosphate, and this is monophosphate because it's lost two phosphates. And hopefully this is a refresher, but we cleave that off and that provides energy. The energy here is causing this reaction. This is the binding site. Here's the binding site for the tRNA to the amino acid tRNA synthase specific for alanine. And then this is the amino acid. Alanine, right? Amino, carbon, and then carboxyl group here. This is the R group. This is the reaction to link them together. All right, so let's talk a little bit about ribosomes. So ribosomes are made up of ribosomal RNA, which we already talked about, and also proteins, right? And there are different ribosomes if you're talking about a prokaryote versus a eukaryote. This S is a Svedberg unit. You centrifuge something and it's a density measurement. So it's a log scale. It's not linear. And so you can't add these together and get, 50 plus 30 is 80 because it's not, because of the way that this density system is measured. So we have the 50S ribosomal subunit, which is the large subunit. And then we have the 30S ribosomal subunit, which is the small subunit. Again, this is prokaryotes. Together, they form the 70S ribosomal complex. There are different ribosomal RNAs for prokaryotes and eukaryotes too. So here we have 16S is in the small subunit, and then there's 21 proteins with that. Here in the large subunit, we have a larger ribosomal RNA called the 23S, and then the 5S ribosomal RNA, and then there's 31 proteins in here. So you should know that the ribosome is a 70S, the combined ribosome is 70S. You should know that 16S and 23S and 5S are part of the 70S ribosome. I don't care, how many proteins there are, but you should know that it's ribosomal RNA and proteins that make up this complex. Okay. So, and the reason that you need to know that is because there are a lot of antibiotics that work on ribosomes and ribosomal RNA that affect their function. And so you should know that what the difference is between prokaryote and eukaryote ribosomes. Okay, so just like in transcription, where we have initiation elongation and termination. We have the same thing in translation. Okay, so this is where it's important for you to put your piles together, right? Remember that transcription is different than translation. The purpose of transcription is to convert DNA into RNA. That's all done in the nucleus, right? The modifications are different, the enzymes are different. And then translation is converting the RNA into a protein where it folds into its proper shape, and that allows for it to have the proper function. So when we look at the ribosome, whether or not it's a prokaryote or eukaryote, it's going to have 3 sites. We have the E site, which is the exit site. That's what that stands for. P site is the peptidyl tRNA binding site. This is where the reactions occur to link them together. And then we have the amino acyl tRNA binding site. Remember, that enzyme is called amino acyl tRNA synthase. That is where charged tRNAs come and bind. So in the A site. So we have the exit site, uncharged. This is where the growing polypeptide chain is linked together. And then this site is where the charged amino acids come in. This is the exit. So we have the small subunit, large subunit. We talked about it. The mRNA goes in between here, shown here, 5' to 3'. It's always going to read that way. We have the exit site, so that the uncharged stick of dynamite is gone. There's nothing on it. It's going to go to the amino acyl tRNA synthase to get recharged. Here's the next tRNA coming in here with its amino acid. The reaction is linked together. So this moves to here, that's cleaved off. This moves to the exit site, this moves to the P site, and then the A site is free for a brand new charged tRNA to come in. All right, so initiation. We just talked about that. Initiation brings the mRNAs and the first tRNA and the small subunit together. So this is a little different. Initiation is different in prokaryotes versus eukaryotes. And so in prokaryotes, the ribosome doesn't look for the end of the mRNA. It doesn't look for the 5' end. It looks for sequences within here called Shine-Dalgarno sequences. Those Shine-Dalgarno sequences tell it where to start reading, and then we'll talk about how this ends in a second. But there could be lots of Shine-Dalgarno sequences in here, and so you would have a lot different ribosomes binding at different places, making different proteins. So in prokaryotes, you could have one mRNA making, let's just say let's say there's I'm going to make this a little longer. So let's say there's another Shine-Dalgarno sequence here. So there's 3 Shine-Dalgarno sequences in here. Ribosome would recognize, bind to that, and then you could have 1, 2, 3 different proteins made from the same mRNA. That's not true in eukaryotes. So in eukaryotes, they find that cap, that 5' cap that we talked about, and they scan until they find the start codon, which is AUG. And this codes for methionine, which it's showing you right here. And then that reads until it hits a stop, which is UAA, UAG, or UGA. These are stop codons. And so in a eukaryote, it reads from the 5' end until it finds the first AUG. This one would also read from the Shine-Dalgarno to find the first AUG, and then it would stop. And this one would also find the first AUG, and so on. The first AUG is the only thing that matters in eukaryotes, and then when it hits the stop, it makes one protein. So you only get one protein from a single mRNA in eukaryotes, whereas in prokaryotes you can get multiple proteins from the same mRNA because of this presence of this Shine-Dalgarno sequence. All right. So there are initiation factors in translation. An example of this is IF1 or IF2. IF stands for initiation factor, and then 1 or 2 is the order it was discovered. And GTP provides the energy. Remember, A and G are chemically similar, right? They're both purines, and so they have the double ring structures, and so because of that, they can react similarly. So you can replace GTP with ATP, and it can provide energy the same way. Just ATP is more common. Okay. So anyway, like we said, initiation is different in prokaryotes and eukaryotes. Regardless of how it is, once the start codon is found by the mRNA, not the mRNA, the ribosome, it's going to recruit, or maybe this has already been recruited, the charged tRNA. It's always methionine. And you look up on the genetic code, you'll see AUG always starts with methionine, which means every protein that you produce starts with methionine. All right, so this is what we just talked about. Here's the Shine-Dalgarno sequence. This is that special sequence, and there can be lots of these in an mRNA. So let me be more specific. Let's say that this is the 5' end, this is the 3' end. Here's a Shine-Dalgarno sequence here. The ribosome's going to bind. It's a lot bigger. It's going to be part of the ribosomal RNA that recognizes this. And then it's going to read, again, in the 5 to 3. It's going to scan until it finds the first AUG. And when it does, it's going to start making that into a protein. So this is the untranslated region, right? Not transcribed, but in this case, translated. And again, you can have lots of Shine-Dalgarno sequences in the same. Again, this is showing that how the ribosomal RNA, in this case specifically 16S, has a complementary sequence to the Shine-Dalgarno sequence. And that's what lines up the ribosome. And again, it's going to scan until it hits the first start codon that it sees downstream, so in the 3' direction. And then it'll start putting in amino acids until, and this is showing you that this is the 30S, right? Which tells you it's a prokaryote. All right, so we're getting a little complicated here, but I don't want you to memorize all this stuff. I'm just showing you what's going on so that it's not such a magic show. So here's our small subunit, 30S ribosomal subunit. Again, we're talking about 30S, so that's in bacteria, not eukaryotes. So we have initiation factors. They're numbered 1. And I don't care that you know these, that these all congregate. Here's the Shine-Dalgarno, here's the start codon, and then here's where the charged tRNA comes in. It's brought in by another initiation factor, and that recruits the large subunit of the RNA. So then when we have the E site, the P site, and the A site here. And these are all released. All right, in eukaryotes, this is different. There's a 40S subunit, right, which is the small subunit. It finds the 5' cap, like I talked about, and it scans until it finds the AUG, the first start codon, right? That codes for methionine always. And so that's how initiation occurs in eukaryotes versus prokaryotes. There's no Shine-Dalgarno sequence in eukaryotes. We don't have a 16S ribosomal subunit. Right. So this is in eukaryotes, right? We have a 40S subunit, so that tells me it's eukaryotes. this E means it's eukaryotic initiation factor 3, eukaryotic initiation factor 4. This is the assembly on the cap. It scans to find AUG. Once it finds it, it recruits the large subunit and everything is released. So I don't want you to memorize all this stuff. I'm not asking you to do that. What I'm telling you is that this process is different, and there are questions in your problem sets for you guys to do this, to convert RNA into proteins. And just remember, you have to remember, are we talking about eukaryotes or prokaryotes? Because it's different. There. Now, I don't want you to memorize this. I'm just letting you know. This is why I want you to know this stuff. Everybody knows that we have lots of bacteria that are becoming antibiotic resistant. And so I just wanted you to know that, you probably learned in micro that most antibiotics work on the cell wall. That's true. But there are other antibiotics that work on prokaryotes, and it takes advantage of the differences in the ribosomes and the ribosomal RNAs, as well as the aminoacyl tRNAs and things like that. So streptomycin inhibits initiation, which causes misreading of mRNA. Tetracycline binds to the 30S subunit and messes up the binding to the aminoacyl tRNAs to the aminoacyl site. Chloramphenicol, that affects the 50S ribosomal subunit in prokaryotes. So it specifically inhibits peptidyl transferase. Did I mention this? I don't think I did yet, but peptidyl did I? No, we haven't done this yet. Okay, so I'm a little ahead of myself. Peptidyl transferase is what is in the enzyme in the ribosome that transfers the growing peptide. Remember, we have the E and the P and the A site. So the A site in the ribosome, large subunit, is where the growing amino acid chain is found. And then the charged tRNA comes in here and has the new. So we have to link these together. So the thing that does the dehydration reaction here is the enzyme peptidyl transferase, which is part of the ribosomal complex. So this chloramphenicol inhibits peptidyl transferase in prokaryotes in the 50S. Cyclohexamide inhibits peptidyl transferase in the 60S. And this is found in eukaryotes, right? So you wouldn't want to give this to a eukaryote. You would kill it because it wouldn't be able to make protein. This binds to the 50S and inhibits translocation, meaning the ribosome can't move. Erythromycin, that's how that functions. Puromycin, that causes chain termination by acting as an analog of an aminoacyl tRNA, and it works in both prokaryotes and eukaryotes. So I just want you to see. I'm not asking you to memorize this. Don't waste your time memorizing this. If you take pharmacology or something, then you might need to know this. But this is the mechanisms by which a lot of the antibiotics that you're familiar with function, and a lot of it is taking advantage of the difference in the ribosomes between prokaryotes and eukaryotes. And that's why I want you to know this. You may develop the new antibiotic that's going to save us all from the superbugs. Okay, elongation. Elongation is basically just the ribosome moving. So this is a still picture of it. It's not an actual video or anything of it, but let's start here, right? So we have the mRNA. Let's say it's all locked in. We have the large subunit and the small subunit locked in. We have the growing chain here on the P site. The charged tRNA comes into the A site. The codon matches the anticodon. We have it here. Peptidyl transferase causes the reaction to link it together. Now we have the growing chain that has been transferred from the P to the A site. The ribosome moves over. So this ribosome is going to move 3 letters this direction. Now this is going to end up in the P site. This will end up in the E site. So here's the growing chain. Remember, this is moved over, 3 letters. This has no more charge on it. This one is this one. And it leaves and it goes and gets recharged. What recharges it? The tRNA synthase that we talked about. Amino acyl tRNA synthase. And then peptidyl we just talked about that. That's what does the dehydration reaction. Once this is done, the growing chain is here. This side is free, and then the next one comes in. And it continues moving on 3 letters at a time until it hits the termination, which is a stop codon. So, again, I don't want you to know. I don't care if you know this. But this is the way that the elongation occurs. So, here's the charged tRNA synthase. It comes in here. This is an elongation factor. That's what that stands for. And then we have a little bit of the wobble here, right, because of the movement. And then we have our GTP, which provides the energy for it. All right. So there are other toxins that inhibit translation, like diphtheria inhibits the elongation factor 2, right? Which drives the translocation in the same way as this elongation factor G works in prokaryotes, and this is in eukaryotes. And if you're interested in this stuff, you might want to go back and look at it. But I'm just telling you so that you know. So here's the elongation factor 2 that we're talking about. And so that it can't translocate, and so diphtheria shuts down protein synthesis, and in that case, it kills you. And it can do this to multiple ribosomes, right? So because it's shutting down all the EF2s, then it's shutting down essentially all the ribosomes. So 1/1,000,000th of a gram per kilogram of body weight is enough to be lethal. There's other things that are used for treating leukemia and other malignant cell diseases that are toxic because certain genes are only going to be expressed whenever you're going through the cell cycle. And we'll talk about that as well. And then termination. So termination is the last step. The ribosome continues until it reaches the 3 letters, UAG, UAA, UGA. Like I said, I'm going to give you the genetic code. So you don't need to memorize these, although it could be handy. So here's the stop codons right here, and I will give you this exact same genetic code on the test, so you don't need to memorize these. You do need to know where they stop, though, that they code for stops. So UAA, UAG, UGA, that codes for a release factor, which is basically a tRNA that's not able to be charged at all. And that has complementary codon, so it has an anti-codon. And that, in this case, there's no way that this can do the reaction to continue this on, and that signals the peptidyl transferase to cleave this off. So instead of doing a dehydration reaction, it does a hydrolysis reaction. This one's released, and everything falls apart. So that's the end of my computer's freaking out. So that's it. So a couple things that I want to let you know about before I finish this lecture is that just like we talked about with the polymerase, once the ribosome is moved out of the way from either the Shine-Delgarno and prokaryotes or the 5' cap and eukaryotes, another ribosome can jump on there. So you can have multiple ribosomes on the same mRNA making lots of proteins from a single RNA. We call that polyribosomes. And then remember the 3-dimensional shape. So the primary structure is just the letters, but that dictates the interactions between the backbone, either beta sheets or alpha helices, and then that in turn dictates the interactions of the R groups, which fold it into its right shape. Now, you can bet your bottom dollar that pharmaceutical companies are really into figuring out the shape of proteins. They spend billions of dollars on this because they want to find the next drug that's going to inhibit something, right? You want to block pain, you need to know the shape of a pain receptor. You want to make a better insulin, you need to know the shape of the insulin receptor, and so on and so forth. So, but here's the problem, is that not all proteins can fold into the right shape by themselves. They need things called chaperones. These used to be known as heat shock proteins, and the reason is because whenever organisms got hot, these got expressed. And well, it turns out what happens when you heat up a protein? You change its shape, and if you change its shape, you change its function. So these heat shock proteins are designed to help hold a protein shape in the correct way. They're almost like molds. And so now that we know that they have more than just a heat function and they're not just heat shock proteins, we call them chaperones. So the chaperones just keep an eye on the protein to make sure that it's folded into the right shape and then it stays in the right shape. We can also do a lot of things to proteins to modify them by other enzymes, and we call those post-translational modifications, which is similar to what we did. We did the co-transcriptional modifications, the cap, the tail, and then the excision of introns and the linking of exons. So we can add sugars, right? In fact, if you look at blood groups, like the ABO blood group, difference between A and B isn't the sequence or the order of the amino acids. It's a sugar, derivative of mannose, that's in a different position on these two. And that makes them different, which causes you to have different blood groups. So we can add sugars and make the protein shape different and modify it that way. We can add lipids, and we call that a lipoprotein. Right? So like if we're talking about HDL or LDL, what does that stand for? High density lipoprotein. Is that cholesterol? No. It's a protein. It says it in its name. This is low density lipoprotein. So that's a protein with a lipid added onto it. We could add phosphates to it. Remember, if we add one molecule to another molecule, it changes its shape. Your body regulates a lot of things by adding and removing phosphates. And remember, I told you guys that I want you to know, well, maybe I didn't, but I'm telling you now, a kinase is something that adds a phosphate. And so that a kinase will change the shape of a protein, and that can change its function. So imagine that you have a protein that isn't in its right shape, right? And it's just sitting in your cytoplasm. It gets a signal, the kinase is activated, and adds the phosphate, changes its shape, and now it's active. That's a really fast response time, as opposed to having a signal and then having to turn on a RNA polymerase, make an RNA, modify it, sit it outside of the cell, have the ribosome read it into the correct protein and have it fold into the correct shape. All of that's done. All you have to do is add a phosphate to it to make it active. So you can respond to your environment that way, just by adding or removing phosphates, adding or removing sugars, adding or removing lipids. We can also modify the length of these chains by removing amino acids from the leading end, dividing them into two or more proteins, so we just cleave them, or we can link them together, right? And that would be a quaternary structure. So all those modifications can occur. And then the last thing we talk about is, remember, prokaryotes don't have a nucleus. So transcription and translation are coupled together. So this is the direction of transcription here. You can see that the growing polypeptides are getting longer, as well as the polymerases, right? So the RNA polymerase and the ribosomes are, as the RNA polymerase is converting DNA into RNA, The ribosomes are on that RNA, making it into a protein at the same time. Because there's no physical barrier like there is in eukaryotes of a nuclear membrane preventing the ribosomes from entering the nucleus or the RNA from leaving the nucleus. It's all in the same place. And in that instance, bacteria are much faster to respond to their environment than eukaryotes are. All right, so that's the end of this lecture. If you guys have any questions, send me an email. And then we'll start into mutation. I'll probably record that lecture on Thursday because I have some stuff going on tomorrow.